Moving on to the third part, sort of how we consider all these traits simultaneously in potato breeding. Pictured here are some potato fruit which look unsurprisingly a lot like tomato fruit and typically have a few hundred seeds in them. So in theory, potato breeding is remarkably simple. I mean, basically you identify two parents that have complementary traits and then you cross them, assuming they flower, that is. And then once we've crossed them, you spend a dozen years sifting through the F1 that you just generated, um, in which are clonally propagated throughout the evaluation, to determine if any of the varieties, any of these new genotypes merit release as a new cultivar. There is no extensive back crossing or any complicated breeding scheme. It's just make a cross and sift through the F1. I mean, conceptually, you can think of it that when you make a cross, you've essentially created candidate new varieties. Each seed is a distinct genotype, and one of those seeds might be a new variety. It just takes a long time to sift through all the seeds to figure out which, if any, merit release as a new variety. So I just said that it typically takes about a dozen years after making a cross to get a new variety. And a question I'm asked so often is, why is potato breeding so slow? And the answer to that, I guess, has three parts. The first part is that it takes about 4 to 5 years to produce enough tubers when you start from true seed in order to do any meaningful yield trial or other large-scale testing. So if you plant a single seed in the soil, you get 4 or 5 tubers. You plant those, and you get 5 to 10-fold as many tubers in the following year, and so on. So the multiplication rate's in the 5 to 10-fold range each year. So it takes a few years to get stuff for testing. Pictured on the right here, I guess a bit of an aside, something that occupies an awful lot of time in a potato breeding program is cutting seed and preparing it for planting, because it's clonally propagated, and our propagules are large. We have to have a lot of space for storing potatoes, and we have to spend a lot of time cutting them and bagging them and getting them ready for next year's planting. When you cut potatoes, of course, you can spread diseases. The worst would be viruses. Well, one of the worst would be viruses. We soak our knives in lime, which you can see here on the right, in between cutting seed lots, so that we're not spreading viruses between clones. Okay, so you spent 4 to 5 years moving from having a single seed to having enough seed for testing in yield trials. The next reason that breeding is so slow is then you have to spend 3, 4, 5 years evaluating potatoes to make sure that the few that you're still evaluating perform consistently. There's really no shortcut for this. It's useless to release a cultivar that yields well 3 years out of 4 and is a bust in the 4th year, because then the grower is going to go bankrupt. When we release something, we have to be confident that it's going to yield reasonably well year after year after year, and the only way to know that is to have tested it in a large number of locations and for a large number of years. No shortcuts here. Okay, so maybe at this point we're up to 8 or 9 or 10 years that a clone's been in the pipeline. Then the last thing that you have to realize that what makes potato breeding so slow is that once we've identified something is good, we have to remove any viruses that may have accumulated or other diseases, and basically begin the seed multiplication process all over again. So at Cornell, I have a counterpart here, Keith Perry, who runs the New York State Foundation seed farm. I send him potatoes that I would like to have virus removed from, because I think it might be a new variety. It'll take him perhaps a year to ensure that any viruses that are present are eliminated, and then from a single in vitro plantlet, multiplication can begin, and that takes a few years until you have commercial quantities of seed available for a variety to be sold. So it's sort of a sum of those three reasons that it takes potato breeding so long, moving from a true potato seed to enough material for testing, then lots of years of testing to make sure a clone can perform consistently, and then finally the whole process of seed multiplication again. In terms of numbers and scales, this slide illustrates a typical progression through the Cornell breeding program, where every year we make 50 to 200 crosses. And then in the subsequent year, we take seeds from those crosses and plant in the ballpark of 20 to 30,000 seedlings, depending on the funding we have at the time by Seedlings. So these are plants that originated from true potato seed, and they produce tubers. And then in all subsequent years, we plant tubers harvested from the previous year's plots for testing and seed multiplication in subsequent years. So when you plant true potato seed in pots, it's really hard to get a meaningful assessment of clone performance. So we don't select very hard on the offspring that are growing in pots. We typically keep about 80% of the seedlings and plant those in the field the following year. We plant in New York 4 tubers from about 20,000 clones each year. And then the way the scheme works is that each year we look at less and less and less clones, but the plot sizes of the clones we're looking at get bigger and bigger and bigger. By having larger plot sizes, we can do more and more tests. We can send the seed to more and more sites for evaluation, in addition to our own evaluation. My program happens to be very interested in developing chipping varieties, so after the first year the potatoes have been in the field, and we reduce the original 25,000 down to about 2,000, we make potato chips out of all of those clones to see which ones chip well, which ones don't. We also care a lot about golden nematode resistance, and so we test stuff once we're down to 200 or so clones, and test clones every year that they remain in the program thereafter to make sure they truly are golden nematode resistant. If it's a chipping clone, we make chips from them every year thereafter too, to make sure we have a good idea of how consistent the chipping ability is. So, oops, I may have gone 2 slides. This is I did. I'll go back. This shows this potato seedlings. You can see there's each of the cells in these seedling trays contains a different potato genotype that originated from a true potato seed. We transplant the seedlings that grow in these seedling trays into larger pots shown here and grow them outdoors, where you can see some indication of some irrigation setup surrounding these beds. At the end of the year, we harvest 4 tubers out of these pots, and as I've already said, we don't practice strong selection at this stage because the performance of potato plants in pots doesn't really reflect what happens in the field. And here you can see some of the tubers that we harvested from pots. We harvest 4 tubers from each pot for planting subsequently, or fewer tubers if that's what we've got. And then in the following year, we plant 4 potatoes in a row. If we're planting white potatoes, we'll plant 4 hills of a white potato, a couple of red potatoes to indicate that's the end of a plot, and a new plot begins. Some more hills of a white potato, a red marker potato, the white potato, and so on down the field. And the colors are reversed if we're looking at reds, where we use white spacers. In the early generations, where we're looking at such high numbers of potatoes, we have to make selection decisions based on a very quick visual assessment of yield and appearance. You can't dilly-dally if you're looking at 20,000 individuals trying to decide which ones you'd like. You just have to take a quick look, make a decision, and move on. In later years, as our plot sizes increase and we have more and more potatoes available for testing and more data available about how a clone performs, the selection process is much more considered and typically takes place with a spreadsheet in front of us and discussions amongst those of us who work in the breeding program. And so this is just a photograph of some of our fields, and you can see here the plot sizes are larger. So these represent clones that are much more advanced in our breeding program. In terms of scale, my program plants potatoes on anywhere between 5 and 10 hectares of land each year. We're not actually limited by the amount of land we have available in my program, but what we're really limited is by how much cold storage space we have. So because potatoes are large, We bring a lot of biological material back to campus each year and have to store it somewhere. And we only have so much storage space that we can keep potatoes in. So it limits the size of my program again um, is storage space and not land. A feature you might notice in this figure is that we plant potatoes, 2 rows of potatoes, and have an empty row in between them. Um, in the empty row, um, we deliberately leave so we can walk the potato fields during the year looking for plants that are infected with virus. If we ever see plants that are infected with virus, and of course we do, we pull them out so that they don't give rise to daughter tubers that can serve as a source of inoculum the following year. So I have often encountered the attitude in graduate students and breeders of other crops that the primary challenge in potato breeding is the fact that it's autotetraploid. And it's true that autotetraploid genetics are much more complex than diploid genetics, but from where I sit, having been a potato breeder for 14 years now, I don't consider the autotetraploid nature of potato to be the primary limitation in making progress. Rather, it's really the high degree of heterozygosity that potato has. So we now know from sequence data that potato has a single nucleotide polymorphism every 20 bases or so on average. And what this means is that every time you make a cross, thousands of loci are segregating. I mean, you'd have an explosion of genetic variation every time you make a cross. And so really what's tricky in potato breeding isn't the autotetraploid genetics, it's balancing all of the segregation that you unleash every time you make a cross. And it's weighing the many strengths and the many weaknesses that each clone possesses to identify something that's better than current varieties. And this really amounts to being at least as much art as it is science, deciding when something really is better, given the combination of traits. You can think back to that list of traits I had before. No potato is ever perfect. It has some strengths and it has some weaknesses, and the ability to discern what's truly better um, is really what separates the potato breeders who are successful from those who aren't. On this vein, we have an annual event in New York called Potato Show and Tell that was started by my predecessor that I've continued. It's been running for a few decades, where I stand up and talk about, in the middle of all these potato crates, the dozen or so potato clones that are currently the most advanced in our program and that what we would like industry feedback on. So I try to do my best to summarize as objectively as I can the strengths and weaknesses of the potatoes that we're evaluating and get feedback from people who grow potatoes, who run potato chip factories, on whether or not they consider a particular clone to be something we should release as a new variety, or whether we should maybe just keep evaluating it, or that we should eliminate entirely from the program. So the business of balancing the strengths and weaknesses isn't something that you can just leave to the breeder with potatoes. It works best when you interact frequently with your stakeholders to get their feedback too. And I probably one of the most important lessons I've learned as a potato breeder over the years is that you can ask growers or industry representatives what they'd like in potatoes, and they can always come up with a list. But in practice, what they want does not always reflect what they've listed as priorities on their list. There are unique combinations of traits, strengths, and weaknesses that they like and don't like, and it's only by having discussions about concrete potatoes with concrete combinations of strengths and weaknesses that you can really begin to discern what they're most interested in. And what they'd like to see in new potatoes. It's a linear list doesn't really cut it. Now, I've said that I don't consider autopolyploidy to be the primary problem in potato breeding, but I have to acknowledge that it is an issue in cases where there is a desirable recessive allele that you'd like to work with in your program, but it's only present at a relatively low frequency in your germplasm or in potato germplasm as a whole. So it's not such a big deal in a diploid inbreeding tolerant crop to fix for a recessive locus. It's much more of a nuisance in autotetraploid potato, particularly for alleles that are present at low frequency. So even if you've managed to drive the frequency of a recessive allele up to, say, 50%, so maybe you have some potatoes that are duplex for recessive allele, and you have a cross where one parent has 2 copies of the recessive allele, and the other parent has 2 copies of the recessive allele. If you cross those 2 parents together, only 1/36 of the offspring are going to be homozygous recessive. And the chances that those offspring have the other attributes you'd like in the potato, not so high. Or another way of looking at it is you got to throw away 35 out of 36 potatoes before even weighing the traits. So this is a big headache if you're considering working with recessive alleles. And for the most part, I personally am not interested in it. There's a second related consequence to this. Let's say you've actually taken all the effort and you've fixed a low frequency recessive allele in some clone and it's successful as a variety. If someone else wants to use it as a parent, and it's truly a low frequency allele, another germplasm, none of the offspring are going to be fixed for that, or homozygous recessive when you make a cross. You'll be fighting that battle all over again with the vast majority of offspring, if not all of them, not being homozygous recessive. I'll find a couple slides changing gears and talking about biotechnology. So, of course, molecular markers have had a profound impact on the breeding of some crops. I guess soybean and maize particularly come to mind, as well as wheat. At the moment, molecular markers are not yet widely used in potato. This is this is primarily due to the fact that very few good markers have yet been developed. The resources, the genomic resources, and the size of the research community engaged in potatoes is less than that of many other crops, even though potato, from a food standpoint, is the third most important food crop in the world. There just hasn't been so much research. That's part of the problem. Actually, that's pretty much the problem. There are nevertheless a few good markers. 2 that I use routinely in my own program are markers that are linked to PVY resistance and gold nematode resistance to simplify the identification of plants that have those traits. I have to say, though, that how you will use markers in a potato breeding program is a little bit tricky, because just because you can identify a plant as having a trait by use of a marker doesn't mean you'll necessarily want to keep that plant in your program. Again, in the end, you have to weigh a lot of traits, and knowing that early on that a plant, is or is not resistant to PVY may not be relevant for you if PVY resistance is not a make or break trait in your program. Because potato is clonally propagated, and we've spent a lot of time developing a cultivar, fixing a unique combination of alleles. It's often easy to think of situations where this potato variety is perfect, but I wish it had just one more trait. Potato happens to be easy to transform, and so if you know the gene that preferred that trait, you can improve potatoes by adding that gene, quite simply. So at the moment, genetically modified potatoes are not accepted actually anywhere in the world, although that could change in the near future. There were genetically modified potatoes in the late 1990s that Monsanto had. They were pulled from the market in 2000, and since then, with the exception of a brief experiment in Europe, which has since ended, they haven't been grown anywhere. Simplot, a company in Idaho, was seeking to change that by putting potato genes back into potatoes and hoping for more consumer acceptance. We'll see how that goes. But in any case, in principle, because potato is easy to transform, genetic engineering of potatoes is sort of an attractive option for breeding if we ever reach the point where they are accepted. In a similar vein, if genome editing using CRISPR or TALEN technologies ever becomes efficient, then similarly I would expect that potato breeding will rapidly change, because if you just like to swap out an allele in a potato, replace it with something different to give it the trait you'd like, that would be exactly what you'd like to do. Again, breeders have spent a long time compiling those allelic combinations in varieties that do become successful, because when you make a cross, it all segregates again. It's often more appealing to think about editing what you have rather than shuffling the dice. And okay, so I guess that's all I really wanted to say on genetic engineering. And this is the last slide. If you're interested to learn more about potato breeding and genetics, I guess the book I'd most recommend would be Potato Genetics by edited by Bradshaw and Mackay. It discusses many aspects of breeding and genetics, and even though it's somewhat old, I still consider it a classic. If you'd like to learn more about potatoes per se, rather than um, genetics, the complete book of potatoes, a book published a few years ago with my father, a colleague of mine and authors, is something I'd recommend. And that, I believe, concludes what I want to say.